All right, so I want to post a quick video overview of this third lab unit for NP2. Good news here, it's only digestive anatomy and histology, no other organ system. But there are a lot of players involved in digestion, as you're learning in the lecture slides. So I just mostly want to run through what you need to know, kind of like we do at the beginning of each of these units. So hopefully by now you've at least looked through some of these and are starting to get a feel for that with this digestive anatomy as you go through the lecture slides. But I want to go over what you need to know, but also there are some specific things in the histology portion that I want to go over as well. So anyway, so just sort of a quick overview of this lab unit number 3 in terms of what you need to know and kind of how I'll test you. All right, so again, the first thing we're going to be looking at is just digestive anatomy. So that includes, the teeth, the tongue, different digestive glands, the esophagus, stomach, large intestine, small intestine, liver, pancreas, gallbladder, et cetera, et cetera. So we just want to roll through all these in terms of what you need to identify. Again, this is nothing new for you guys since you've been through this before. Everything you need to identify are in these boxes, right? So you want to go through and identify these different regions using the pictures provided, just like we've done previously. So in this lab manual, you're going to start with the buccal cavity or the oral cavity, right? You're within your cheeks, the different boundaries of that. And then you're going to move down to the pharynx, identifying those different areas of the throat. And this picture will definitely be one where you can get a lot of information in terms of things that I'll ask you. So that's a really nice picture in terms of identifying and finding those structures. This one as well for a lot of the oral cavity stuff like the vestibule and the palatine tonsil, the fosses and the soft palate, hard palate, et cetera, uvula. Roll through all those. So I would use those pictures to identify those structures. And then you move on to the salivary glands. And fortunately, we have a really nice model of this lab, which I won't be using, of course, just be using the pictures on the next slide. So there are 3 parotid, submandibular, and sublingual salivary glands, which you can see nicely in this picture here. So make sure you run through that. Then you want to move on to the teeth. And the teeth, mostly what you're going to be identifying are the different types, so incisor versus molar versus canine or cuspid, et cetera, and then the different regions of the teeth. So to do that, you first want to look at the different regions and tissues, which you can see nicely on this picture. This is a great photo where you can see all the different regions, the parts, crown, neck, root, but also like enamel, dentin, gingiva, so forth and so on, periodontal ligament, et cetera, et cetera. So use this picture here to identify those different structures. And then in terms of the different teeth, incisor versus cuspid versus molar, et cetera. This is a nice picture because you can see the different root structures, but then this also just shows them within the roof and the bottom of the mouth. I actually would use the adult teeth picture, so it would probably be this one and the next one in terms of identifying the different teeth. So for instance, if I had this picture and said, what tooth is this? You'd write third molar. You don't need to know the years of eruption. Again, that's more of a topic that we'll look at in lecture, which you may have come across already. Then on to the tongue. Really what we're looking at, again, are the things in these boxes, primarily the lingual papillae, which you can see on this next slide. So anything related to the tongue, in terms of the structures listed would be used on this picture. So that's pretty straightforward in terms of those different structures. So again, if I had a picture of one of these, you'd have to identify that as the fundiform papilla. But I would use that photo or that picture there. In terms of the esophagus, you have different regions of the esophagus that you're going to be identifying. We'll do histology here in a minute. You probably won't be able to see the different sphincters or the esophageal hiatus. Those are probably more model-related items. But you do want to be able just to identify the actual esophagus in terms of, where you'd find it. So if I used a picture to identify the esophagus, I would use that. Then you move on. Esophagus, of course, dumps into the stomach. In the stomach, there are different regions like the cardia and the fundus, pylorus, etc., the different curvatures and sphincters, all of which you can see. This is a really nice illustration for all of those different features of the stomach. You can also see the end of the esophagus there, which is nice. And you can see the beginning of the small intestine over here, the duodenum. So that's a really good picture to identify a lot of different structures. Small, of course, attaches to the stomach, and so we'll look at the different regions, duodenum, jejunum, and ileum, which you can see nicely on this picture. So this is a really good one of the different regions of the small intestine. That, of course, attaches to the large intestine, and so you have a lot of different features you want to identify in the large intestine. This is a great picture for that, where you can see a lot of the different structures from the ileocecal valve to the cecum, to the ascending, transverse, descending colon, all the way to the rectum. Different flexures, right and left flexures, which you can see in those pictures as well. So that's a really nice picture for all the different structures of the large intestine. This is a great shot for the illustration for the rectum and the anus and the sphincter muscles, which you have to know as well. So that's a good picture for that. Next is the liver. We have the different lobes, ligaments, and other structures, cystic duct, hepatic duct, etc. And so we want to identify those. This picture definitely shows a lot of those structures, which you can identify. The next picture as well has different lobes and different hepatic portal areas, different ligaments. So that's a really good picture. This is also a really nice shot, not only of the gallbladder and the liver, but you can see a lot of the ducts in this region here. So make sure you look at all those as those go into the pancreas. Next one you want to look at is the pancreas, the different bile ducts and the connections into the small intestine. You can see a lot of the pictures here. So this is a great illustration for all of that. Identifying those different structures. Again, the common bile duct coming from the liver is also joining with the pancreatic duct as they dump into the duodenum of the small intestine. Again, we discussed that a little bit more in lecture, but that crossover will come there. Here's another nice picture of that bile duct coming together with the pancreatic duct as they dump into the duodenum. All right, so that's it on the identifications of the anatomy. Last part of this is related to histology. And so we'll look at the histology of these different organs. You have 5 different histological sections you want to look at. And so we'll just roll through those real quick. First one is the esophagus. And each time as I go through this, I'll test you. I'll have you identify, which organ this tissue sample comes from. And then the other thing that I would ask you would be to identify some features related to this tissue. So this is the esophagus. So if on the exam there's a picture of this histological section, I'll say identify this, what organ is this tissue sample belong to, and you would write esophagus. Next thing then, I could ask you some features about that. And so the first is the mucosa or stratified squamous epithelium. Is this section right here, right? You guys are familiar with stratified squamous epithelium by now, and you can see the multiple layers of stratified epithelial cells in that location. Mucosa is just beneath that here. And then finally, the muscularis externa, or smooth muscle, is all the way down in this region. So if you pull those up a little bit closer on your PowerPoint presentation of this lab manual, you should be able to see those details. So those are the things I would want you to identify with the esophagus in terms of the histology. Next is the stomach. Stomach, again, tell me, correctly identify which organ this tissue sample belongs to. And then you want to look and identify specific features as well. I could ask you, what region is this here? And that is the simple columnar epithelium. You can also see these goblet cells, these white cells that you see sort of scattered amongst the simple columnar epithelium. These goblet cells secrete mucus. As you're learning in lecture, mucus secretions are very important in terms of coating the lining of the large intestine or the stomach, as well as other regions of the digestive pathway. But it also helps, provide a lot of protection from the acidity that we see in the stomach. So those are the goblet cells. And then this opening here, this big opening is what's called a gastric pit. That opening would lead further down, which you really can't see in this picture, but it would lead further into what's called a gastric gland. But so this opening, if I ask you what this opening generally here is, that's what's called a gastric pit. So those are different things you want to identify within the epithelium or the histology, of the stomach. Then you want to move on to the small intestine. Small intestine, this is an illustration of the small intestine. And some features you want to be able to identify first is what's called the plica circularis. Plica circularis is a fold within the small intestine that doesn't go away, which you can see here. It's made up of several villi. And so, like, for instance, one of these structures here is what would be called one villus, singular villus collectively. All of these are called villi. And all these come together to form one plica circularis, and there's another one next to it, and so forth and so on. On the surface of these villi, you have simple columnar epithelium and goblet cells, just like we saw in the stomach. Those are a little more zoomed out, but those would be the surface of the intestine. And then finally, the muscularis externa as well is smooth muscle, which you can see in that picture there. So those are the different things I'd want you to identify for the small intestine. The liver, this is a nice shot of the different lobes. So like, for instance, the one lobe of the liver. And within that, you have all of these simple cuboidal epithelial cells, which help filter our blood and remove toxins and other types of things. Those cells are typically referred to, these simple cuboidal epithelial cells are referred to as hepatocytes. And then you also can see nicely this portal area where vessels come into and then are distributed through those simple cuboidal epithelial cells. So those are the different things I'd want you to identify with the liver. And then the last one is the pancreas. The pancreas, if you recall from AMP1, we talked about the pancreas sort of having this dual function, organ system function from an endocrine standpoint, but also from a digestive standpoint. And if you recall, the endocrine cells are called islet cells. Islet cells are the things that produce like insulin and glucagon, which help regulate blood sugar. But the vast majority of cells within the pancreas are what are called pancreatic Acini cells. And these are the cells that you're seeing scattered amongst all of this tissue sample are these Acini cells. And then you can also see nicely a branch of the pancreatic duct to allow for secretions. Ultimately, they're going to be released into that location. So those are the different things you'd want to identify on the pancreas. So anyway, so that's it on the lab manual number 3. Pretty straightforward in terms of identifying different illustrations that I've scattered throughout this presentation, but then also make sure you have all the different histological sections down. All right, that's it.